Now, we've got a great session coming up, three speakers again before we have a chance for lunch. Please do use the breaks to catch up with people if we don't get a chance to ask your question or it occurs to you later. Our first speaker in this session is Stephen Frankenberg from the University of Melbourne. Now, Dr. Stephen Frankenberg is a zoologist and geneticist at the university in the School of Biosciences. His early research was focused on embryonic development of mammals, especially marsupials, and expanded into reproductive biology of vertebrates more generally. He has a lifelong interest in evolution and ecology, and then moved into genetic engineering as a way for solving problems in conservation biology. So he's now worked on a whole bunch of really interesting projects, things like protecting northern quolls from cane toad toxin, protecting frogs from chytrid fungus infection, and eradicating invasive vertebrate pests. So incredibly relevant for today as well. Stephen, thank you.
Thanks, Tim. Okay, so I'm going to talk about some of the work that we're doing in my group at Melbourne University today. I'll just start off with, obviously, these are sort of stats that you've heard. I think this chart here down the bottom left, it's the third time today you'll have seen that. We hope somebody's keeping a tally for the number of times we see that today. But one interesting thing about it is that deer are sort of not represented there as in the top 10 species for threatening species, other species. And I guess maybe the reason for that is that it's just nowhere near the population number that it's going to get to. So you can see that the increase in numbers over the past few decades is still on that exponential curve like you see in the early COVID days, and there's just no sign of flattening there, and that's quite frightening. So, I mean, we really need the conventional methods, traditional methods for controlling their numbers, like using lead bullets, but we really need a silver bullet really in the longer term if we're going to address this, because we don't really have the resources or the capacity or the political will at the moment that can actually suppress those invasive species, let alone eliminate emergent species, let alone eliminate the established ones. And one of these emergent ones that I also will mention later on is the smooth newt that's established in Melbourne. So this silver bullet that I believe is likely to be, very likely to be, solve a lot of our invasive pest problems in the future, not all of them, is genetic biocontrol and particularly gene drive. So the amount of investment so far in developing gene drive is a very tiny fraction of the cost of invasive species at the moment, and it is increasing at the moment. There's been some significant boosts to funding opportunities lately, and hopefully that will increase a lot more so that we can get there. So gene drive is a particular type of genetic biocontrol, one that is able to sort of spread exponentially through a population in a sort of self-sustaining kind of way. So it works basically by when you sort of normally inherit a copy of a gene, so you have two copies of every gene in our genome, one you inherit from each parent, and it's a 50% chance of inheriting each of those copies of those genes to the offspring. In the case of a gene drive, ideally that biases that inheritance to up to 100%. So even if there's a cost to that gene drive, then it'll keep on spreading through the population regardless. And so the extra trick is if you target a female fertility gene with a gene drive, then it'll cause the females to be infertile, but the gene drive will keep on spreading through the population via the males regardless, until you end up with a population of only males and no fertile females, and the population crashes. And so this is potentially amazing technology, a solution to our invasive pest problems. Other advantages is that it's self-sustaining, so you only have to introduce it initially and it will just keep on working by itself. And it's also very humane, of course, because they're animals are essentially copulating their way to extinction, essentially, by that process. So success in developing this technology will firstly be in the gene drive design itself, to get them to really sort of copy efficiently and spread. And the other challenge, which is one of the main sort of things that we're focusing on in our lab, is simply the ability to produce animals carrying gene drives, the logistics of doing that, which is not a trivial thing in all these sort of non-model species. So I'll try to not go too much into the technicality of how a gene drive works, but I'll just sort of give a basic overview. It depends on this molecular tool called CRISPR, which you might have heard about. It was sort of first developed about 10 years ago. The people who developed it got the Nobel Prize for doing it, and it's essentially a molecular tool that allows you to sort of very efficiently edit a genome in a way that sort of was never really possible before. So it uses this enzyme called Cas9, it's the key enzyme, and it basically allows, using a guide RNA, it's a small molecule that sort of guides the Cas9 enzyme to a particular part of the genome precisely and makes it cut the DNA at that point in the genome. So it creates this double-stranded break in the genome. And so if this is on the top left there, you've got a gene drive construct, which has got your guide RNA and your Cas9. It cuts that's the wild type or the normal version of the gene and creates that break. And then the natural DNA repair mechanisms in cells will actually then use that gene drive as a sort of a template to repair that break in the DNA. And by doing that, it actually creates a second copy of the gene drive. And so then you have two copies, and then all of the offspring will inherit that gene drive. So that's the basic mechanism by which it works. So in our lab, part of our work is optimizing that design of the gene drive to make sure it works efficiently. And so this is an example of, this is what my PhD student Clancy is doing with his work, using the zebrafish as our sort of easy-to-use vertebrate model to develop the technology. He's created this sort of split gene drive for biosecurity reasons. We don't want to be creating gene drives, fully fledged gene drives in the lab in case the zebrafish get back to the foothills of the Himalayas where they come from originally and wipe out all the zebrafish. That we have the two components of the gene drive in different parts of the genome, so if the animals sort of get out, then it's not going to sort of cause a problem. So basically, you can see the glowing red fluorescent embryo on the right, showing that he's sort of produced these constructs or produced these animals that he can then cross with each other and test different components of the gene drive to see which kinds of designs allow them to spread more easily through a population. So I won't go too much more into that side of it, of our work. The other side is simply the logistical challenge of getting gene drives into these non-model species. So if you want to sort of make a transgenic animal or knock a novel DNA element into a genome of a species, it's quite easy to do in something like a laboratory mouse, but to do it in species, our invasive species like carp and toads and fox, etc., it's not a trivial task. So for species like carp and cane toad, you can do this just by injecting your CRISPR construct. So we're also using CRISPR here as a way of getting these gene drives into the animal. You can do this by just injecting the eggs that you've collected, thousands of eggs. You can just sit there injecting away, and then you screen for the fluorescence that tells you that you've actually managed to get that in. So you can see we've had some really great success recently. There were teething problems for a while in getting all the processes working, but now we've managed to get our first carp knock-in embryos to show that we can get any construct we want basically into an embryo, and then we can grow those embryos up into adults ultimately and breed from them. And we've recently done that also with cane toads. So the green fluorescent embryo there is showing one that is showing a knock-in into a fertility gene, in fact, of cane toad embryo. And now we're basically raising those toads, or toadlets they are at the moment, up to adults so we can actually breed from them. So it's easy to do, well, not easy, but that's how we do it in carp and toads. For mammals, we can't collect thousands of eggs at a time and sit there injecting them. So we have to have a different process. So the way we're doing it is actually to set up cultured cells, get cell lines from these different species, so get pieces of tissue and then grow up the cells in a culture dish. And then we do our genome editing in the culture dish. And then we use a technique called nuclear transfer to basically to create an embryo from those cells that will then have those inserted constructs. So this is basically the same technique that was used to produce Dolly the sheep a few decades ago by cloning, where you use cells that are growing in a culture dish to basically create an embryo, and then you put that embryo into a uterus and you've got your animal. Beg your pardon? We're going to do hopefully a better job than Dolly, yeah. So this is an example of a colony of rat stem cells from the black rat that we've recently created in the laboratory as well, where we can do these edits and then use that for nuclear transfer. And so that whole pipeline that we've sort of developed, this is how we envisage it would work for something like a fox. So we've already got our cell lines established from red fox, and we've shown that we can sort of knock in any construct we want. This is a construct that's been knocked into the Y chromosome of these fox cells using CRISPR, glowing red there to show that we've done it. And then in the future we can use, once we've got a gene drive in there, we can use those cells for nuclear transfer. So we need the eggs that we use the nuclear transfer from. So that could either be from ovaries from vet clinics, from dogs, and try and do it in a cross-species kind of way. If that doesn't work so efficiently, then we may have to rely on getting ovaries from scavenged tissues from hunted foxes to get ovaries and to get those oocytes that we take out the genome from the original egg and then replace that genome with our new genome that's got the gene drive knocked into it. And then we can grow those up to an embryo and cryopreserve them and wait till we get the funding to then sort of transfer them to the uterus of a fox in a fox captive bred colony to produce our gene drive foxes. So I've talked mainly about the sort of techniques that we use for gene drives for established pests. We've also got the problem of newly emergent pests like the smooth newt, which has established a population in southeast Melbourne, and it recently appears to be spreading it as well. So it may become hopefully not the cane toad of the south. So I think it's something that needs to be nipped in the bud fairly soon, in case it does become a much bigger problem. So we don't have any funding for this, but it's kind of a fun and interesting project, and a good challenge is to try and develop a quick self-sustaining biocontrol, because we don't need a self-sustaining one with a small population. We can just release animals that have been genetically modified to basically suppress the population there in the short term. With repeated releases, you can eventually eliminate that population. So it's not a genetic modification in the normal sense. In fact, it's a sort of a trick that's called a Trojan Y, where you have XY, you produce embryos that are genetically male, so they have X and Y chromosome, and you treat them with estrogen as they're developing, and they'll develop into XY females, and then you cross those with normal XY males, and you produce a quarter of those offspring will be YY males, and they're the ones you release into the environment, and all of their offspring will be male, because they'll all inherit a Y chromosome. So you keep on repeatedly releasing those season after season, and eventually you'll wipe out the population. So this is the simple technique we're trying to use to address that problem. So caveats. Obviously, there are plenty of caveats when you're talking about gene drive. One of them is the emergence of resistance. So people always say life finds a way. That you'll get resistance emerging to these gene drives. So in fact, there are ways around making sure that this resistance doesn't arise. So you have multiple guide RNAs that sort of all target a whole range of sites within, this is in the CRISPR mechanism, that sort of make sure that there's not going to be sort of resistance emerging in that fertility gene that you're targeting. I sort of won't get into the technicalities of that, but basically gene drives are not pathogens. So it's not like viruses and things that the species have evolved, their immune system has evolved ways of sort of recognizing these pathogens and getting immunity and developing resistance to them. It's not the same kind of thing. Eradication may not be, maybe incomplete due to you've got sort of patchiness in the environment, so you've got this sort of effect, I think Ben might talk about this more later, that it's difficult to completely eradicate the population because the wild type animals keep on escaping from the gene drive. But even if in a self-sustaining way you can maintain the numbers or down to sort of maybe 10% of the population level that they would otherwise be, this makes an enormous difference to native species and allowing them to recover. The risk of introduction to non-target populations, so for example if cane toads were to hitch a ride back to where they are originally native in South America, and we don't want to be responsible for that. Australia is a world leader in biosecurity as well, so I'm sure that we would have a responsibility to make sure that what we're producing doesn't get out of Australia, and we're sort of in the best position to do that because of our expertise. But anyway, even if that were to happen, which I think would be a slim chance, it's actually easier in principle to to engineer resistance to a gene drive than it is to engineer the gene drive in the first place. So you could actually have a resistance engineered in the non-target population in South America that would make sure that if gene drives got there, it couldn't possibly wipe out the population. So there's sort of an extra backup involved. Obviously, this is gene drives are something that spreads through breeding. So when you've got animals that are very long-lived, I think Ben will talk about this later as well. It could take 100 years or so with these larger species like feral cats and foxes before it eventually sort of wipes out the population. For small, fast-breeding animals like rabbits, it should happen nice and quickly, just within a few decades. So, to have the most efficient impact with the gene drive, you have to sort of couple it with, invest heavily in lots of modelling. There'll be, lots of work that will be required from ecologists and population modellers, and captive breeding of having large facilities that breeding up these gene drive animals and having strategic releases in the environment and eDNA, environmental DNA, to detect where animals are existing and where the gene drive animals are spreading to will be very important in this process. So there'll be a lot of extra expertises that will be required coupled with this kind of a program. And as the saying goes, the best time to plant a tree is 30 years ago. So even though it's a long time scale for these things, it really needs to be done as soon as possible. And this is just a list of all of the people who have been very helpful and collaborators as well in all of these projects. Thanks very much.
A question? Yeah, Ewan, get going.
Well,
I can just talk. I've got a
yeah, I can hear you.
That's brilliant work and it's so exciting. So I guess two questions. The first is, when's the first release and what species? And
depends on funding.
And the second one is, excuse my ignorance, but obviously I know we've been concerned with things like, cats and, getting into the, felids in, Asia and so forth. But that's obviously when you've got an invasive species that potentially can co-occur with wild native species. Mm-hmm. But when you've got the situation where we have things like deer and rabbits, which obviously have both, they're pets, they're commercially produced, as long as there's not contact with the wild, am I right that it's all okay?
As in, will the
physical contact?
Yeah. Will people's pets be affected by it?
Exactly,
of course, yeah.
Or their production animals in the case of deer. As an example.
Exactly. Yeah, I mean, people who own sort of domestic cats as well, shouldn't be allowing their cats to breed with feral cats. But if they did, the worst thing that's going to happen are the offspring. The offspring are going to be sterile, and so people will still kick up a fuss about it, I'm sure. But with feral cats, if you've got sort of, programs to try to eradicate cats from the environment, but you've got this constant seeding of new cats, new populations. So I have thought very heavily about this sort of thing, and I do have sort of designs that I've sort of thought about that would be, getting this legislated in the domestic cat industry would, be a hurdle in itself. But there would be ways of, I think, having regular licensed breeders who can only breed animals that have sort of inbuilt gene drives. So the system that I've sort of developed is where you give them a drug that allows them to be bred, but they're fundamentally gene drive animals. So if they don't have that drug that allows you to breed, they won't be able to breed. And then the offspring will carry that gene drive. So if they then, become stray cats, they're not going to establish a new population. So there are ways you can engineer a solution to that problem. That'll take a long time to sort of, develop those and get them, acceptable to society. But I think we'll get there with this sort of stuff.
That's brilliant, thank you.
Another question? Yeah, down the front here.
Well, probably a comment, but exciting technology, wow. I wouldn't, when you mentioned the caveats there, I wouldn't underestimate the antis. I can still remember when the ANU did that research many years ago, and the antis blocked it and won the argument.
Yeah, I think it'll require small wins at first, and example on offshore islands where you're sort of just controlling a rat population that's eating the seabird eggs, and those sort of those small wins people will see that if you've got genetically modified wildlife, the sky doesn't fall in, it's fine. the ugly animals that people, like cane toads or whatever, people won't mind that as much. And I think the same thing happened with GM foods. A few decades ago that was evil, and now it's kind of much more accepted. So, but yeah, it's obviously a consideration. Yeah.
Yeah. Look, thank you. And I should have not warned really, but it's going to be some complicated science in this session. But I think it's fascinating to hear the detail, because you'll be some of the people that can talk to others about, yes, this is what they're doing, but they're also looking at the problems and dealing with those. And I think it's the ethics plus good science to back up those decisions, which is what you're doing, and it's the nuance in this once again, which is where we started. So look, thank you very much for that.
Oh, sorry, just one quick question for me, if it's over here.
Oh,
Sorry.
yeah. Sorry.
Yeah, sorry. Hi, Lockie from Parks Victoria here. Just a quick question. Is any of this sort of gene harnessing technology being used in countries, say, New Zealand, that have more progressive policies than, say, we do here?
Aggressive
Aggressive
in the
or
sense of
progressive?
so New Zealand has sort of shunned gene drive mainly into this. It looks like they might be starting to shift and starting to fund some genetic biocontrol research, but initially they were totally shunning it. But no other countries really for that. I mean, it's Australia and New Zealand are the two countries in the world that have the obviously the most to gain from this kind of thing. Yeah.
Thanks for that question. Thank you. Fantastic. Thank you. Our next speaker, Dr. Alex Fournier-Level, is a senior lecturer and group leader at University of Melbourne. Background in grape breeding. For his PhD, research focuses on, again, genetics of plant adaptation to the environment, in this case, for the good and the bad, as he says. He's got strong expertise in weed genetics, working on agricultural weeds, particularly herbicide-resistant annual ryegrass, and ecological weeds such as common ragweed. His lab has developed new modelling techniques and uses genome information to support decision-making for Australian biosecurity, as we heard a bit about before. So thank you, Alex.
Thank you for having me. That's really a pleasure. Great introduction, Tim. Thank you, Bart. And really thank you for your presence here as a community. I think we can make a difference. And we've heard a lot of great talks about animals, and I wish to emphasize that, unfortunately, in the context of plant science in general and weed science in particular, we lag behind because our species being less charismatic than beautiful days, we're suffering from lack of engagement, lack of support, and chronic underfunding. It's also the case for animals, but unfortunately it's even worse in weed. So I'm a senior lecturer here at Melbourne Uni, and I wish to disclose a conflict of interest. I had blackberries for breakfast. So why do we use genomics? Because I engage with Annelise explaining that one data point is going to cost you probably $1,000. So the eye sparked. Yes, it's still cutting edge, extremely expensive technology we're using, but there's a few things we can do with genome technology that you could not do with anything else. And in the context of plant invasion, in particular, we've got challenges that are slightly different from that of animal science. Typically, we can think of identifying species. When you see a deer, we may discuss which species it is, but when you see blackberry, I dare you don't know which species you're talking about. We don't, Bart, I'm not sure even you know. So there's hundreds of species, and they all have slightly different features. So identification is the first step in biosecurity when it comes to weed, and that's not a trivial effort. Next, we've got the issue of tracking movement. You have beautiful cameras that do a fantastic job at spotting big, bulky animals. My blackberry seeds flying high in the sky in the bum of some random blackbird. Tough to track, hey? And finally, where we really add value is because we're touching the genome, as in the underlying cause of biological functioning, we're actually tracking the system, we're trying the mechanics, the functional dimension. We're not only giving you a place and a time, we're also telling you what the organism is doing. So if you think in late term what genomics can do now, and I'm going to introduce you to a couple of examples where we're actually implementing biocontrol and biomonitoring using genomics, not just having a wishful foresee of what could happen in 10 years, is think of this beautiful experiment carried on ants where a very docile graduate student would paint the back with unique barcoding, you know, strap by strap. Can you imagine doing that? Well, that's what genomics does for you. We already have a lengthy long band with mutations that literally barcode the uniqueness of each genotype. So it's not at the individual level, because your identical twin by definition would have the same barcode as you, but still you can discriminate between different biotypes, between different steps in evolution, and this is going to be critical for what I'm presenting here. So this is what we believe is a valid model for biological invasion. I'm purposely drawing a beautiful square cell with a vacuole in the middle, so animal biologists don't get confused. Here is typically an invasive species invading. You've got its genome that is currently all gray. Upon arrival, if it manages to hit the first step of an invasion, and that's really not happening. In Australia, we're getting 1,000 species introduced every year. Luckily, only a very tiny fraction becomes invasive. That's an opportunity for us. Unfortunately, the guys who stay are really the obnoxious ones. And what happens generally in that background is that we have the emergence of an adaptive mutation. So my first line on my barcode, here I'm depicting it in purple for the sake of the presentation. And this new mutation is going to propel the species by making the first adaptive step in the Australian environment. Identifying niches that it can colonize. Right now it's restricted and you don't see it by eye. You don't see it coming. It's a plant, you don't care, it's not charismatic. But that's unfortunate because within that background, very likely at that stage, there's going to be a new mutation that this time is not going to be adapting the plant, but it's going to modify the breeding system, the breeding capacity, and dramatically expand the range. And at this stage, it's too late. So let me show you an example of the work we're doing in common ragweed. So who in the room has heard common ragweed? Some specialists. So it's coming to us. It's the most allergenic weed in the world. 25% of the global population, and we're very equal. There's no ethnic difference. 25% start to sneeze. And students, at the beginning of the project, immune to the weed, by the end of the year, generally, they started to sneeze. It's globally invasive, which is bad, but it's an opportunity. And currently in Australia, has a limited range. So it's time for action. So I'm presenting one of our plots off from Coffs Harbour, where we're working with a local land manager, and typically the land manager has fenced off a plot of land for us to conduct some survey and analyze the demography. And so that kind of stresses the need for a partnership between the academia and the land manager, because there's no way I can identify population and do a good survey without this local connection. So this is really priceless. Where I can take it from there is actually go back to what I'm training students for and good at, which is large-scale data analysis, leveraging all the records ever placed on the map for ragweed. And again, this relies extensively on the goodwill of some enthusiasts. And on that space, botanists have always been more committed in putting observation in repositories. So this is really, the community dimension is really there, even if stakeholders with bigger purse have no interest in us. Day-to-day wanderers do care about maintaining their ecosystem pristine. So I'm leveraging all these accessions. So you've got the knots representing occurrence record, and the crosses represent occurrence record where we did analyze the DNA. We do have the genome sequence for these plants. So what we can observe is that the plant is native to North America, has invaded the biggest chunk of Europe, really going from warm to really cold climate, so we're really concerned about its potential to adapt to virtually anything. And as you can see, in Australia, it's still quite restricted to a smaller bound of the east coast, and we wish it to continue like that. So what do? We analyze the occurrence, and we're trying to identify the environmental conditions under which the plants thrive, based on all these occurrences. Leveraging big data, what we're capable of computing is the niche, the theoretical niche, the space in which these plants thrive. So first, very easy finding. You've got the 4 different ranges, as in global ranges in the green background. You've got, within the triangle, the superimposition of what is the suitability in North America, not the suitable, the current range in North America, the current range in Europe, and the current range in Australia. So with respect to land use and land cover, you can see that those spaces more or less overlap, and all the type of land cover that are currently suitable for the plants are already covered. And this stresses one thing, and that's fundamental in weed biology, is weeds are not that much of competitors. They're not that much of adaptive beasts capable of withstanding any stress. They mostly respond to perturbation. So any land clearing will result in recruitment of weed. Weeds are super pioneers. Unfortunately, the Australian environment is based for stasis, is based to maintain itself over multiple years, potentially without rainfalls. Most of the grass systems are capable of withstanding 0 rainfall over 2 years, to only get back to active growth on the 3rd year. The problem is when you're clearing the land, you giving the opportunity of things that run short term to take over an empty niche, and this is where the problem comes. However, one thing that we noticed is that when you focus on the climate variables, so the climate, precipitation and temperature mostly at the location of occurrence, here you can see that the niche in yellow, that is the current Australian niche for ragweed, is very small compared to the other niches, and that means unfulfillment. That means in Australia we've got a great potential for ragweed to take over to the entire available climate space in Australia, and we've got such a wide variety of climates in this country that we have reason to be concerned about explosive radiation of that species. So let's look a little bit under the hood and see what the adaptive potential of this plant is. And I'm not going to run into too much detail, but that means that at the genetic level, we've got 3 big families. Cluster 1, cluster 2, cluster 3. Sorry, we're not very original with the naming, but in short, you can see that, and again, my apologies for the color code, but in Australia, we've got a paucity of representative of cluster 1. So there's a family missing, and we really hope that they're not going to do a cousin gathering over Christmas. But we've got this lack of cluster 1, and what we're capable of modeling based on our genetic data is that if we take the current Australian range, and if you were to be quite permissive with your biosecurity by introducing source of ragweed, sorry, that looks ugly, source of ragweed, you're going to have to peel your eyes, from this cluster 1, as you can see in the red dot, I wish it was bigger, you can see that the south of Australia is at risk of invasion by specifically sourcing North American line, so you get a vessel coming with whatever forage crop is coming from the US to feed sheep. You end up specifically with cluster 1, this one family that is missing in Australia, and you're at risk of having your species all of a sudden invading the most of Victoria and South Australia. Don't tell me why this one is working. However, interestingly enough, if you do the same with a European introduction, then cluster 1 no longer matters. We don't care about getting the family 1, but if you're re-supplementing more cluster 2, more cluster 3, you end up with the invasion potentially going northwards. And in the context of cluster 2, really taking over the northern tip of the Arnhem Land, which are places that are notoriously difficult to access, let alone to manage. So this is really to show you that if you just do biosecurity based on origin, you're completely missing out the genuine risk. But if you add genetic information, then as you can see, differences are quite dramatic and worth catering for. In the context of climate change, when I was doing research in climate change, we were always saying that genomic allows you to give more hope, more potential for adaptation. Unfortunately, in the context of biosecurity, it's also the opposite, but that should make us dread even more invasion, because there's a lot of things that happen in the genome, the evolutionary potential, that is currently not catered for in ecological model, and I think we should work on that. Now I'm going to spin to another project where we're directly supporting farmers and ecosystem or agrosystem profitability with Lolium rigidum as the costliest weed in the country. It seemed like a good idea to introduce a forage crop that was really drought tolerant and really annual to make a quick cash buck by harvesting in season, storing and feeding the sheep later in the season. Unfortunately, we didn't know by then, like often in biosecurity, that we would let in the most potentially most herbicide-resistant weed ever seen. It's resistant to pretty much anything that was thrown at. And what we can do by sampling extensively is retrieve the genetic information from a quite extensive sampling around Victoria and a little bit beyond, match it with the herbicide-resistant profile for 10 herbicides we've been testing in the lab, train what we call a genomic prediction model, which is leveraged in plant breeding all the time, but to my understanding, never in weed science. And this model allows us to predict patterns of resistance. And the goal is to be able to support farmers who can directly ship a sample. We're going to extract the DNA and analyze the data instead of running 10 herbicide tests that are labor intensive, condition specific. The genome doesn't change. Genome is quite set. So this information is valid today and tomorrow across any herbicide or any biological feature for which we've gathered enough information. Currently, our model does okay. So this is response to glyphosate. So as you can see, or may not see, in the top right, oops. In the top right corner, we identify really well resistant weeds. Unfortunately, we tend to be a little bit unspecific for susceptible weeds. So we can, in the left-hand side, you see the points are a little bit all over the place. However, what is pretty cool is that there's nothing at the bottom right, which means that when you've got something resistant in the paddock, I do know that it's resistant. I'm not going to call it susceptible when it's resistant, which is really what matters in this context. So now to conclude, I'd like to bring it back to blackberries, because that's the topic. In the context of blackberry, genomics would be fundamentally important to identify species. We are talking about aggregates, not even species complex. We don't know really what's in the field. From what I can tell from yesterday, a quick analysis, 78% of the records are not ID'd at the species level. They're just genus, nothing more. And we do have some native blackberries we have to deal with. So it would be a little bit of a pity to just blanket bash everything without necessarily having good information. The other idea is herbicide is still at the forefront of management practices and shall remain, unfortunately, for a long time. But we need to do better with less. And typically, the DPI New South Wales has already shown that blackberries susceptible to rust are susceptible to specific classes of herbicide. But this is species specific, and we need species information. So here we stand, and we can support a better way to control and manage weeds at the local level. Tracking the movement, that's great, but I don't think it can be any worse. As you can see, observation from the last 4 years tells you that I do believe that, unfortunately, the niche is fulfilled in the context of Australia. However, still, analyzing the dynamics of this population would be fundamental and could allow us to actually measure the outcomes of our management practices. Because of the barcoding system, I can see the shriveling of genetic diversity within that species, and without genetic diversity, there's no adaptive potential, and potentially we're getting closer to eradication. Finally, herbicide resistance is something we do particularly well in Australia and in my lab, and making sure that we are not creating a new monster is important by monitoring resistance level. So I know I'm running over time. However, I wish first to acknowledge the lead researchers from my group. So Andika is a graduate student. Jeff is a postdoc that since has moved to become a research scientist at Agri-Victoria. The support from the Australian government. So we started by being supported by DAWI, then it became DOR, then it became DAF, but it's all the same people. They're lovely. And we also receive funding from the GRDC. We are the Adaptive Evolution Lab. You can follow us on Twitter, and I'm leaving the website address for if you want a little bit of a better picture of what we're doing. Thank you very much.
Thank you, Alex, and thank you for speeding up a little bit there at the end. That's wonderful. Any questions, please, for Alex? I was actually pleased to see taxonomy appearing as a taxonomist by trade, but blackberries are notoriously difficult to sort out, aren't they? They're hybrids. I suppose one question I might have was around, once you understand that taxonomy, and I guess it applies to the rusts and some of the pests, but does it help you understand the population dynamics at all better as well?
Absolutely, and mostly when it comes to hybridization, both potential, realized, or prospective. Like there's notorious barriers to hybridization linked to the number of chromosomes, and across the different species of rubus, you may have different number of copies. You know, we've got 2 copies of every chromosome. Such is not the case for every species. Plants are notoriously easy and permissive when it comes to the number of chromosomes. Not everything can hybridize with another. But this is something that generally generates a superweed, typically in the context of a sea rocket invading our seashores. We created, well, we let a superweed being created by hybridizing a maritime version, very bulky version of the sea rocket with what had the first pioneer species. And so we had Cachile edentula, first sea rocket, landing, then being able to hybridize with Cachile maritima, and now we have a problem.
Cachile is a sort of beach weed, some of you would know. Yeah, we'll take one question, then we'll move on. Thanks. Well, that's a good comment. Yeah, Lynn, do you want to add something? Yeah.
The weed CRC, which I don't think is still, it's not. They were actually running Blackberry ID, and which some of our members went to, and lots of community people. So there is a potential to, maybe not through them, but somehow to do that again.
Absolutely, this is me whinging at the beginning about chronic underfunding. It's not that there's no great initiative, it's just that they last while they last, and unfortunately we forget about them, but the problem hasn't been solved.
No, that's right, but there's potential to
and the technology is ripe now. We're delivering solution, as you can tell from my herbicide-resistant type of emergence. I can point you at spots in the landscape where risk is there. Yep. We can exactly know, typically in the case of ragweed, what we need to track and what we need to look for. It takes money.
Yeah, but we've all got a role in that. I mean, I think. I don't know how you actually do that when there's no money at government level, but then, you have to try that, don't you, to this sort of...
I'm not going to stop.
No.
Maybe we should leave that as pose that question, is how we get better support. Is that on the same topic, or...? We'll take one more, one quick question.
It's in relation to you saying that the community gives you the data what databases do you know, we've got the iNaturalist for the plants and things, but there doesn't seem to be a specific one for weeds, and we've got our EVBC, which is, you know, the atlas at the moment is down more than it's up. So what do you look for when people are inputting? What databases do you refer to?
For observation, the Atlas of Living Australia, that's our main source. And hopefully, based on last comments, I think their funding system has been secured for a short foreseeable future. I'm hoping to integrate molecular genetic data into the ALA. So we don't know how, but we've got talks to make sure that this is being integrated at this level, and that's our main source. But we're relying on calling people and landowner and land manager. So you get the grad student designing its survey along the coast. They're getting themselves an Airbnb and they're contacting the local managers. So unfortunately, we're not at the stage where we've got a federated monitoring system. It's really a tip on the shoulder and ad hoc communication.
Thanks for that. And look, I think iNaturalist and Atlas of Living Australia should be where all these things are recorded. I mean, as much as possible with a lot of projects, we're trying to incorporate them back into that. So there's one, I know, but one source of information that can be reliably checked and all that kind of thing. So in the end, iNaturalist is probably the place to start. Thank you, Alex. Our final speaker for this morning's session is Ben Phillips. Professor Ben Phillips is from Curtin University, a professor there in the School of Molecular and Life Sciences. He's a population biologist. I'll let him set up while I'm doing this. Thank you. With a background in ecology and evolution, particularly interested in spatial processes and how they influence population and evolution and evolutionary dynamics. Began life as a field biologist, so he's actually been out there seeing the plants, which is not always the case, and animals for some biologists, and has morphed into a modeller, which seems to be the trend here. He's interested in developing models covering population and evolutionary dynamics and applying these to real problems in agriculture, health and environment. Thank you, Ben.
Thanks very much for the invitation to come here today and talk. I thought I'd expand a little bit or extend a little bit on Stephen's talk, talking about the promise of gene drives for controlling and eradicating pest species. The promise is that we can have this super Mendelian inheritance. Sorry, is that better? Testing. We can have this super Mendelian inheritance process where a genetic element we put in can drive itself through the population, and we can use it to skew sex ratios, and we can use that to cause a population to go extinct, right? So the promise is that something like this happens, right? That's a simulation of a gene drive doing what we would hope a gene drive does, right? So it's an incredible promise that is before us. When I first heard about gene drives about 10 years ago, my initial reaction was one of mild horror. When you put humans behind the wheels of just about anything, surprising things can happen, right? And I'd come from a background of invasive species, and so I'd been thinking about, so the cane toad is an excellent example of a self-disseminating biological control effort that has gone quite horribly wrong, right? And gene drives are a self-disseminating biocontrol effort. I've come around dramatically on my initial perspective on that. I think gene drives are potentially very valuable. I'll talk about that in a second. The first thing we need to, I guess, after I thought I was mildly horrified, the second thing I thought is that actually they probably won't work, right? And the reason for this is, yes, you can drive these things into populations, but there's still, we don't get around Darwin, we don't get around natural selection. They're still pushing against natural selection, and they can push to a certain degree. But but when you're driving something extinct, you're obviously potentially creating very strong natural selection. And if there's any variation for that to work on, potentially your gene drive gets driven extinct by natural selection. And the obvious and the strongest problem at the time, about 10 years ago, was the issue that the gene drives themselves would create resistant alleles, right? And so in the process of that copying that Stephen talked about, you can get poor copying happening, and you can get alleles that pop up that are resistant to the gene drive. As soon as you've got that, natural selection then has a toehold, right? And what will happen is that resistant allele will run to fixation instead of the gene drive, and so the gene drive fails. And so my initial thought was, oh, we don't need to worry about this too much because it won't work. Of course, what's happened since then is people have come up with a range of really quite sophisticated and clever ways to get around that, right? So the technical advances have now gotten to the point where that problem is solved, I would say. There's still other places that evolution might find a toehold, and those will be specific to the particular species that you're working on, right? But certainly I think the largest problem of resistant alleles popping up through the process of the gene drive itself, I think that problem is solved. So they probably will work. The next problem is a more surprising one, and that came along a little bit later, and that's the issue of space itself, right? So we're trying to put these things into space. They're moved around by dispersal, and they're propagated through by reproduction. What happened in that last simulation I showed you is that the gene drive spreads from the centre of where we introduced it, and it creates that empty space behind, which is the population going extinct, and it drives forward. This I wonder if I can get this pointer to work. No. Anyway. This figure on the right here shows you that process in one dimension, right? So you've got a gene drive that's been introduced on the far right. It's that blue curve, and it's been pushing up through the population and driving it extinct. And you can see that vacant space that's been created behind the gene drive. That vacant space is kind of constantly in the process of being colonised. And so it's possible that gets colonised by a wild type rather than the gene drive itself. And so you can get, and when that happens, you can get a situation emerging like this, where the gene drive is doing its thing, and then you can see it gets colonised by a wild type behind there. And so there's a large part of parameter space, and that's a range of dispersal values and reproductive values where this kind of dynamic appears quite naturally in the simulations and would definitely appear in nature as well. That can cause, you can see it takes a lot longer for extinction to happen, right? And in some cases, extinction doesn't actually happen. You can get this running on forever. And in other places, you can get what's about to happen here, which is actually the drive goes extinct and fails altogether, right? So this problem also seems like quite a fundamental problem. Strangely enough, having gone from, I think gene drives are mildly terrifying, I'm now working towards solving this problem with Charlie Robin over at University of Melbourne. I think we have a trick that we might be able to deploy that could potentially stop this from happening altogether. So we're back into that situation with the first simulation. So again, it's another technical hurdle, or it's another hurdle to deployment, I guess, that is looking like it might be able to be beaten through technical advances. And this is happening very, very fast, right? These things were kind of only dreamed up really, a decade ago. Problems been raised, problems been solved, problems been raised, problems been solved. So the technical development's happening really fast. One thing we won't be able to get around is the issue of time, right? So because these things are spread through reproduction, generation time is going to be a fundamental limit to how fast we can get these things to work. There's some really nice work done out of the University of Adelaide, Macy Brown and her collaborators, showing how long it might take to eradicate a population of 200,000 of each of these particular species. And you can see there's some surprisingly big numbers there, like 250 years, right? Now, the thing you'll notice also is those long tails running up. Those ones are caused by that chasing phenomenon that I just showed you before, right? So if we can solve that chasing phenomenon, we can bring those tails right back down to, we can make them all look like the rat example rather than the fox example. So by solving that chasing problem, we can potentially bring those numbers down quite dramatically. But we will still fundamentally be left with the issue that it's going to take time for these things to propagate and spread and do their job, right? That's okay. It's a fairly low-cost option, right? Once you've released them, it's not like you necessarily have to manage them ongoing, but it's not an instantaneous quick fix. So that's a fundamental constraint. I was not the only person to be mildly concerned about the possibility that gene drives presented, right? In 2016, one of the early proponents of gene drives, Kevin Esvelt, pointed this out, and he was saying, well, you know, the situation that Stephen showed before, right? We make this bit of technology that wipes out cane toads in Australia. Well done us. Too bad if it gets to Brazil, where toads are native. That would be really bad because we would potentially be wiping out a species in its native range rather than in its invasive range. And Esvelt's point was that these things are designed to spread. And so we have to be really, really careful of them because they can, you know, it's like a spark in a dry grass field, right? This thing will take off. And so he was pointing out that, quite rightly, I think, that we need to be quite cautious and careful about developing this technology because surprising things can happen, right? As Stephen pointed out, it's possible to engineer, I guess, genotypes, if you like, that are resistant to the allele, right? So it is possible to create populations or to generate populations that are immune to the gene drive. So that's one way we might do it. But almost only a year after this paper came out, someone else pointed out that there's another strategy that we can use to actually physically contain drives, right? And that is, it's quite a little bit complicated, but these things have a threshold. Or that you can build drives that have a threshold. They have to get up to a threshold frequency in the population before they will increase in frequency, right? So in this particular example, we have a drive that has a threshold of 0.5. So half of the population has to carry the drive. If anything less than half of the population carries the drive, you get this situation on the top where actually over time the drive actually just slowly goes extinct. If, however, you get that population above 50% of the population has the drive starts to increase in frequency and then it starts to spread through space, right? So we can engineer these things to have these thresholds in them. Those thresholds are really important because, or what that generates is a thing called a pushed invasion wave. They've got lots of interesting properties, but one of them is that they tend to stop at barriers, right? So the top example is the kind of spark in the grass field type of design, a pulled wave where it will grow from very low frequencies, right? You've got this thing spreading through space. It hits a barrier, a place where there's very low population density. The drive decreases in frequency, but it pops through the barrier, and because it can grow from very low frequency, it just takes off again. On the bottom, we have this situation where it has to get above this threshold frequency. It's moving through space. It hits a part of space where there's a low density of organisms. You get the drive moving through that barrier, but because it never gets above its threshold frequency, it's just constantly going extinct on the other side of the barrier. And so these barriers can, and so we can engineer drives that are physically containable, right? And if someone picked up a couple of cane toads that had this drive and they took them to Brazil and let them go, nothing would happen, right? Because you haven't been able to bring the frequency up high enough for the drive to propagate. So we can design these things to be physically containable. The other way we need to contain them, of course, is within the species that they are targeted at, right? We were talking before about, this is out my back door one day. I just walked out and they're not even in the same genus. Anyway, very, very hopeful, amorous male frog. So the issue is, right, if we design these things and they're supposed to, sit inside a cane toad or whatever, and that species hybridizes with something else, we can potentially have escape across species, right? And that is something that is quite a concern. I think in Australia, for many of the species that we might look at, that's less of a concern because, for example, cane toads, there is nothing in Australia that cane toads can hybridize with. There's nothing in Australia that newts can hybridize with. There's just nothing that's even remotely closely related to them. So it's a good test case, or it's a good use case. People are also working, I guess, on the design technology type side of things to try to keep them contained within the species as well, but that's a harder problem. I just want to talk quickly about social license, and I guess what I'm getting at here is we are getting to the point where it's possible for us to design drives that I think many people would feel were reasonably safe and reasonably containable. And if we deploy them in the right kind of circumstances, I think, in fact, it is entirely possible that we can design drives that are safe and effective. The next problem, of course, is social license, whether people are going to think that that's okay for us to do. I think that we have become inured to the fact that a lot of the things we do to control and manage invasive pests, or invasive species, are actually quite awful. We do an awful lot of shooting and killing and trapping and poisoning has all sorts of nasty off-target effects. Think about the declines of insects all around the world, right? We've become inured to it because we don't have a better choice, right? If we were in a world where we were using gene drives for this kind of stuff all the time, and someone said, oh, look, I think we should switch to shooting, killing, trapping, baiting, everyone would say, well, that's a stupid idea because it's way more expensive and it's unethical, right? So I think the social license here can be built around that idea. Instead of doing awful things to creatures to try and control them, those creatures just don't exist, right? That's what a gene drive is doing. It's just, they don't exist. So from an ethical perspective, it's substantially superior, I would say, if we can get them built safe. So I think that's a conversation that we need to have and that can go forward from here, right? Actual licenses are going to be quite a challenge. This is a draft policy guide that's just out now. I think public comment is still open. Basically, it's saying you have to, a proponent that wants to release a gene drive in the wild needs to demonstrate it's a clear public good, that all the risks have been acknowledged and mitigated. You then need to get approval from the Office of Gene Technology Regulation, that's fine. But they also say you also need to get approval from every federal and state government that might have an interest, which is all of them, right? And all of that falls on the proponent. All of that falls on the proponent, right? So it's going to be harder to get up than the voice. Maybe that's an exaggeration, but there's a large amount of lobbying. There's a large amount of work that would need to be done to actually get approval to release one of these things in the wild with the way the current regulation is set up. And it's set up in a very much a we don't want to take any risks kind of way, right? I think that conversation needs to continue, and I think we need to come up with a slightly better solution than what's currently on the table there in terms of licensing these things and thinking about ways that we might gain approval to release them. So I think that's a conversation that needs to move forward from here as well. So in summary, I think there is really great promise here. The technical hurdles are falling very, very quickly. This is a decade-old technology, and there's people around the world really interested in it, and the technical hurdles are moving very quickly and being solved. I think we're really close to the point where we can have drives that are pretty damn safe. That said, surprising things are always going to happen, and so it's always going to be a risk-benefit argument. Generation time's a hard constraint, nothing we can do about that. And we certainly need to have these conversations with the public about the potential benefits of this technology compared to what we're currently doing. I think that's the way to have that conversation. And I think we need to think about the regulatory landscape going forward. It's definitely true that we need a regulatory landscape. We definitely want that. It needs to be rigorous and thoughtful and careful. But in a conservation setting, proponents aren't going to have the millions and millions of dollars that they're required to meet the regulatory hurdle that is currently looking like it's the hurdle that's there currently. So I think we need to think about that as well. And that's it. Done.
Thank you, Ben. I know we're eating into lunchtime, but I will, if there is a sort of burning question, yeah, please, just here at the front or the middle.
Yeah, hi, thanks for that. That was really interesting. Public versus private, do you see an issue with the potentially in America, I guess, of gene drive technology falling into private hands and causing issues?
Yeah, I think to some extent this comes back to Alex's talk. This technology is becoming increasingly accessible, and I think there is definitely a risk that it will be developed and accidentally or otherwise deployed. And so we need to be ready for that, because I think it's a very real risk. So biosecurity is increasingly not just, you know, we're already feeling overwhelmed by all of the species that are arriving, but now there's going to be genes arriving that we need to pay attention to. I don't know if that answered your question or not, but yeah, I think we need to potentially step up the kind of monitoring we're doing to look towards the genetic level.
Yep. Okay, thank you. And the other part of my question, completely unrelated to the first one, from a land care point of view, we're finding that the community is getting increasingly involved in lots and lots of things that were previously, I guess, the realm of the scientists and things. From a practical point of view, what does the gene drive release process actually look like? Like, can you imagine a scenario where community groups like Landcare are handed a couple of cane toads?
Yeah.
And out they go and do the release? Is that something that's possible, that community can support this in a really, really big way?
Yeah, so in some sense that last step is ludicrously simple, right? You just need, and if it's a threshold drive, you need to introduce a large number of whatever the organism is to bring the frequency up in the population, and that's it, right? So you don't have to go out there every year. I mean, you might want to go out and monitor it to see where it's going in space and time and all that kind of stuff, and make sure it works. But the deployment of this thing, once they're made, it's the same as bringing cane toads to Australia and letting them go. They just do their thing, right? So yeah, so I think yes, you have to, but you also have to think about where in space you do it and how many you bring, because it'll happen more quickly and more effectively if you scatter it through space instead of just letting it go here and let it spread across the country, right? So in that sense, I think you know, land care and so forth would be a really powerful conduit for getting them out. Yeah.
Okay, thank you.
Thank you. It's a great question, by the way. Really quick, back and front. Yeah.
Sorry, just a quick question. I'll preface my comments by saying that my postgraduate studies was in biotechnology, and we discuss some of these implications quite extensively. Is where you take this technology and bring it into the human population to eradicate disease and social strains, if you like, as to control the way population looks like in the next 100 years. And that was pretty scary. Will that be the next step from animals to humans?
Not from me. Yeah, I'm not even sure what to do with that question. It's Margaret Atwood has some interesting ideas. Yeah. But again, I mean, you think about it as a potential risk, but then humans have a generation time of 20 plus years, right? So even if someone somehow snuck a gene drive into the human population, we're going to find it pretty quick, and we're going to know about it, and it's not going to have gotten far before we've worked out how to deal with it. So I think, in some sense, we don't need to worry too much about that.
Sharon, did you want to add something?
I just had I guess it's kind of probably hasn't really been touched on in the three presentations today, and something I probably struggle with, which is putting, and we fund and do a lot of gene drive work, putting a lot of an invasive species back into the environment before you get a return, and I guess the impacts that will cause when you've got 100-year plus, and obviously your threshold example, what sort of work's been done around that side of it? Like, are we, like, to get to the point where they are going to be beneficial, are we going to do more damage to the environment in the meantime? And I know that's a difficult question. I just want it to be on the radar and the agenda.
Yeah. No, I don't think I have an answer to that one either. I think a lot of things, for example, I can talk about it from a cane toad perspective. Most of the damage has already been done. And so you can introduce a whole bunch more toads and you're not going to make much difference. I don't know how true that is generally, but yeah, it's definitely interesting.
I think it does. It sort of brings us back. Probably, I thought the social license point you raised before was really interesting to think about that. And, the shooting, the poisoning, all that stuff we do, and then weighing that up against these other risks. So it's risk management, and making that decision. And it does remind me very quickly that people often say to me, or have said, if everyone had electric cars, now we suddenly said, why don't we bring in petrol cars where you stand next to somewhere and you pour this liquid fuel into your car and then you light it, and wouldn't that be a great idea? It seems like a weird thing. So it is spinning that back round to see what the risks are in reality. So thank you very much, and thank our three speakers for this morning. Fantastic.